Good afternoon. It's always a pleasure to be the first speaker after everyone's had a couple of beers. So the title of my talk here is Discovering Cellular Defenses. As Matt pointed out, I'm a cell biologist. I study disease at a molecular level, and the diseases that I do research on are lung diseases, pulmonary diseases, specifically a disease called chronic bronchitis. And chronic bronchitis is a disease where the mechanism, as it says on the slide, where the mechanism goes bad. The mechanism is mucus. We all know about mucus. We all get mucus when we have a cold. And it's normally a nice defense mechanism in the lung. But when we have too much mucus, we get a disease. And diseases like chronic bronchitis, and even as Matt talked about asthma, can be characterized by too much mucus in the lungs. So what I'm going to do is give you a little background. I'm one of the fortunate scientists who's been able to take a discovery that I made in my laboratory and take it to the point where it's now poised to become a drug to treat patients who have this disease. So I want to take you sort of through the path that got there and use those scientific types of concepts to try to expand into the world at large. Say, what can we learn in the laboratory that we can use as a microcosm of life in general? So these are the lungs. We all have them. So I want everybody in the audience to just take a deep breath. I'll do it with you. And then exhale. And what just happened? As you can see from this diagram, there's a bunch of pipes lead from your mouth to your nose that get down into the lungs. This is the trachea. You can all feel your trachea right here. It has rings of cartilage surrounding it that protect it. And as you go down deeper and deeper into the lungs, the pipes get smaller and smaller. When the breath that you just inhaled gets down into the lung tissue, the oxygen gets into the bloodstream, and at the same time, the blood, which is carrying a lot of carbon dioxide and other waste products, releases that into the air. When you exhale, you're getting rid of all your waste gases, and you do this 60 times or so per minute. Now, I was going to try to compare the respiratory airways to a tree, but Roy Underhill here had an actual tree, so I'm not going to do that. What I want to do, though, is take one of these lungs here and do a cross-section. Say we just take a knife and cut right through there and show you some of the structures that I'd work with. So this is what it would look like. This yellow material here looks like a sponge would be the actual lung tissue. These are blood vessels, these dark stained regions, and these are the structures that I'm interested in. Those are the pipes that lead down to the lung. These are the bronchi, the airways. And if we were to take a look at the airways at a microscopic level now, remember I'm a cell biologist, I want to see what kind of cells are there. We'd see the top surface here. These are the cell types that line the bronchi. And I want to draw your attention to these three cells here in cross-section. These are called goblet cells. Why are they called goblet cells? We don't have to use our imagination too much to say, hey, they look like a wine goblet. I was going to say a beer bottle upside down, but I wouldn't do that. And rather than being filled with wine, these are filled with granules of mucus. And what happens is that these goblet cells discharge their mucus whenever there's any sort of stimulus on the cell's surface. And so we always have a layer of mucus coating our airways. Well, that's great. Mucus is a large polymer. It's a thick, viscous, sticky, I can put the word disgusting in there since we've all eaten, a material I think that we're all familiar with. Evolution of mucus. Why would I have a slide like that in there? Well, let's think back thousands of years to our ancestors walking around caves or plains or wherever they were. And what were their biggest threats to life, other than saber-toothed tigers or wild animals attacking them? And that was when they inhaled material, it was filled with bacteria, or they would inhale material filled with bacteria or viruses or other microbes. They couldn't go to if they got an infection, they couldn't go to a drugstore. They couldn't deal with it. So what was developed or evolved is this system of defense against bacteria and microbes. That's called mucus. The cells see, hey, there's a bacterium there. Let's put a distance between the bacterium and the cells with this thick, viscous, sticky stuff. That way, it won't attack the cells and won't hurt us. Works great for thousands and thousands of years. Enter the year approximately 1800, and what happens in the world? It's an industrial revolution, coupled with increased use of tobacco products. So now, instead of breathing nice air in a cave or a plane, suddenly we're breathing air filled with particles and pollutants and smoking cigarettes at the same time. So our airways are getting hit with all these different materials. Our airways can't think, cells can't think. So they just say, oh, there's a bacterium there, let's produce more mucus. So get more and more and more mucus. Mucus clogs the airways. As a result, you get diseases, industrial disease. Remember they saw from Dire Straits in the 1980s. A good example of that is chronic bronchitis. It's just a good defense mechanism gone bad. And what happens is, if you look at the normal bronchus on the left, normal airway, the one on the right here is just filled with mucus. So when we breathe in, we can't get a good air in. When we breathe out, we can't get a good air out. There's millions of people in the world today that suffer from bronchitis, and that was the focus of my research. So here we have an actual airway from a human, and these are the actual goblet cells, and those little dots there are actual mucus granules. And the question is, how can we stop these granules from being secreted? How can we treat people with bronchitis and stop all this excess mucus from getting into their lungs? So now the goblet cell is represented by this rectangle. It's filled with these granules. This goblet cell gets hit with the stimulus. The mucus gets released. We want to keep that mucus in there. Okay, now we switch to an oval to represent the goblet cell. And there's a stimulus at the surface. There's only a single granule there representing the mucus. So we get a stimulus. The granule gets secreted. How does it work? What's in that question mark, that black box? What kind of molecules are involved? If we want to cure a disease, we have to understand how the normal process works. So what's the normal process? What kind of molecules do we see in there? Well, 15 years or so of research got to the point where my laboratory discovered that the protein involved, the molecule involved in regulating mucus, is a protein called Marx protein. Now, I know a lot of you are sitting in the audience and say, oh, that's awesome. I remember the Marx, from the Marx Brothers, night at the opera, day at the races. Anyone under 40, of course, doesn't have no idea what the Marx Brothers are. That's Harpo, Chico, and Groucho, thank you. I'm over 40. Marx is an acronym for myristylated alanine-rich C-kinase substrate. I won't go into anything scientific other than to say that's what the molecule looks like. And when I first started doing this, there was no way to inhibit Marx function. So my second big finding in my laboratory was we developed a way to inhibit Marx function using this peptide. That's not a misspelling of the men's room. The man's actually stands for something, and the first MA stands for Marx. So we published a number of papers related to Marx protein and how it actually blocks mucus secretion. And it does so in cells in culture. It does so in a number of different animal models of disease. Trouble is, and any scientist will tell you this, a lot of things work in a test tube, a lot of things work in a petri dish, a lot of things work in mouse models of disease. If we were trying to cure mice of asthma, we'd have this not 15 years ago. Trouble is, what happens with human patients? So I wanted to take this discovery and see if it could help patients with disease. Now, I can't just go out into the hallway, find somebody coughing and say, I got some drugs for you, why don't you take those? And the reason would be that I'd be arrested and put in jail with all the other drug dealers. You have to go through a number of different steps to get to the point where you can do studies with humans. You have to do a test to get approval from the Food and Drug Administration and then go on to do clinical trials in humans. We all know clinical trials are important. We see television commercials proven clinically. Well, yeah, unless it works in patients, it's of no value unless you're a mouse. So we found to our delight that indeed this peptide did work in patients with chronic bronchitis. I want to show you one slide from that clinical trial here. Just try to make this as simple as I can. The purple, well, the x-axis here are the percentage of patients who responded in a significant way, whose lung function got better after taking the medicine, the drug. This is the days they took it, for 49 days. HNS is the placebo, stands for half normal saline, and the drug is given at that concentration. That's shown in purple. So there are two things I want to point out here. One is the placebo effect that you've all heard about is alive and well. And you see about 15 to 25% of the patients get better just from inhaling saline. Mine can do a lot of things to the body. Second thing I want to point out is that people taking the drug get better and better and better over a course of time, to the point where day 28, half the patients taking the drug actually showed a clinically significant improvement in their lung function. This is, I would say, the height of my research. I was very, very excited to see this. And hopefully we can get this out as a drug. Okay, let me try to bring this now into a different perspective. People have a concept, an idealized idea of how these systems work, how scientists do things. They think of a hypothesis, they test the hypothesis, they get the results, everything's cool. Well, couldn't be further from the truth. This is really the way it works. You get a hypothesis, test it like I said before, but the trouble is the results don't fit the hypothesis. So you tell a graduate student or whoever's doing the studies in your laboratory, go back, do it again, you must have done something wrong. They go back, they do it again, same result. Now you get really excited, you're really not doing this right, get back and do it, because my hypothesis couldn't have been wrong. They go back, they do it, same result. So what do you do now? You can beat your head against the wall, you can try as many different ways to test the hypothesis as you can, or you can take a hint from what you see and say, wait, maybe you're doing it right. Here's a raise. I want you to stay in this laboratory, don't be going to anyone else's laboratory, and change your hypothesis. I think the key to being a good scientist is changing your hypothesis. Like it says here, every hypothesis I ever had in the last 20 years has been wrong. It had to be changed. What I want to do to bring this into a realm of life is to say, well, how is science, how is the stuff we do in a laboratory relate to a bigger sphere of life? In science, you have to be prepared to change or alter your hypothesis. The results drive it. You have a hypothesis, you test it, the results aren't what you think, you change it. The results drive it. In life, your results aren't going to change unless you do things differently. If you're trying to reach a goal, if you're trying to accomplish something, you need to change your hypothesis if, in fact, you're not accomplishing that. Albert Einstein has a quotation, and you can look it up on Google if you don't believe me, and it says, insanity is doing the same thing over and over again, thinking that this time the results are going to be different. And if you do the same thing over and over again the same way, the results aren't going to be different. If you're trying to get to C, and you got A plus B equals C, and you keep doing A plus B and it doesn't equal C, at some point you have to say, well, that's not working, let me change that. Let me try D and F to get to C. And you can take that paradigm and move it into any sort of professional, social, school, work, any situation you have. To get the results, sometimes you have to change your approach. Let me give you a better example from my life. This good-looking guy here was a freshman at Carnegie Institute of Technology in Pittsburgh in 1962. I wanted to be a civil engineer. In 1966, I got this letter from Carnegie Institute of Technology. You don't have to read the whole thing. You can see the word drop over there, and that's university talk for flunk out. So yeah, I flunked out of engineering school in 1966. My hypothesis was that I'm going to be a successful civil engineer. Trouble was, the results didn't bear out that hypothesis. I wasn't a good civil engineer. I I wasn't a good civil engineering student. Now, I could have tried another school, I could have tried different ways to be a civil engineer, but it wouldn't have worked out. The conclusion that I came to, and obviously there was a lot involved at the point that time, was maybe you should change the hypothesis, maybe you should try something else. So, new hypothesis is, let me try biomedical, see if I can be a good biomedical researcher. So instead of getting letters that drop out, I now get letters like this. This is an award that I won in 2005 from the Board of Governors from the University of North Carolina. This is it's called the Merit Award from the National Institutes of Health, which essentially funds my research for 10 years without having to do anything. When I say not having to do anything, obviously I have to do something, I just don't have to keep asking for more money. So, I'm a university professor. Whenever I give a lecture, I want my students to leave with a take-home message. So what's the take-home message here? And I think it is that although we're not all scientists in a laboratory, you can take the laboratory as a microcosm of life, and in that regard, we are all scientists of life. When reality doesn't fit what you think it should be, when reality doesn't equal the ideal, you don't have to do the same thing over and over again. You can change. It's all right to change. It's all right to, it's not a disaster if what you think is going to work out the way you want it to work out doesn't. You don't have to take it as a negative, you can take it as a positive. It might even be as simple as, using an analogy, if a light bulb burns out, go change the light bulb. If your idea doesn't work out, change your approach to it. The idea is okay, but the way you're going about getting it done might not be. And I guess the real take-home message here is that the results aren't going to change, your approach needs to, and it's okay in life or in science to change your hypothesis. Thank you.